Your ability to absorb and use creatine is partly written in your DNA. The SLC6A8 gene, which encodes the creatine transporter protein, has known variants in the human population that affect transporter function, and these variants explain some of the inter-individual variation in how people respond to creatine supplementation. The creatine transporter is not a fixed, identical protein across all humans. Like most proteins, it exists in multiple variants, called polymorphisms, that arise from single nucleotide changes in the gene sequence. Some of these variants change the shape of the transporter protein in ways that affect how efficiently it binds and moves creatine across cell membranes. Research has identified specific SLC6A8 variants associated with lower creatine transport efficiency. Individuals carrying these variants show reduced muscle creatine uptake in response to supplementation, slower loading rates, and potentially lower maximum creatine stores even after extended supplementation. These individuals sometimes describe themselves or are described in studies as non-responders. The term is imprecise but reflects a real phenomenon. Some people supplement consistently and see less improvement than others, not because of training errors or dosing mistakes, but because their transporter is less efficient at the molecular level. The functional consequence of a less efficient transporter plays out differently depending on context. For muscle uptake, a less efficient SLC6A8 variant means slower loading from blood to muscle. Your muscle creatine stores rise more slowly and may plateau at a lower level. For brain uptake, the CRT1 transporter involved in crossing the blood-brain barrier has separate variants, though the brain transporter genetics are less well characterized. The severe creatine transporter deficiency syndrome, an X-linked condition caused by loss of function mutations in SLC6A8, causes profound intellectual disability, absent speech, and seizures in affected males. This clinical extreme illustrates how critical transporter function is. The mild variants that produce suboptimal but functional transport still allow normal physiological function, while complete loss of function is catastrophically disabling. How do you know if you're a transporter variant carrier? Without genetic testing, you can't know directly, but you can observe your response to supplementation. If you've supplemented consistently at 5 grams per day for 8 weeks, trained hard, and haven't noticed the expected improvements, no extra reps, no change in interset recovery, no scale movement, a less efficient transporter variant is a plausible explanation. Some individuals with suspected transporter efficiency issues respond better to higher doses, taken with carbohydrate to maximize insulin-facilitated uptake, sustained over longer periods. Others may find the benefits simply more modest regardless of protocol. For these individuals, the cognitive and health benefits of creatine, which operate through mechanisms that may be less transport-dependent, may be more accessible than the performance benefits, and worth pursuing regardless. The genetic angle on creatine response has a broader research context worth understanding. SLC6 A8 variants are not the only genetic factors that influence creatine's effectiveness. AGAT and GAMT, the synthesis enzymes DIKE, also have population variants that affect how efficiently your body produces creatine internally. Variants in AGAT that reduce its activity result in lower endogenous creatine production and lower baseline muscle creatine. This means a larger saturation headroom and potentially a stronger response to supplementation. Conversely, individuals with more efficient AGAT activity produce more creatine internally and may start closer to their ceiling before ever supplementing. The combination of synthesis enzyme variants and transporter variants creates a range of baseline creatine phenotypes across the population, from individuals who naturally run close to full saturation to those who are consistently operating well below their ceiling. Genetic testing for these variants is not yet routinely available in consumer contexts, though some research panels include these markers. For practical purposes, the observable outcome, how your performance changes with supplementation, remains the most accessible indicator of where you fall on this spectrum. An 8 to 12 week standardized supplementation trial with careful performance tracking before and after is a reasonable and accessible phenotyping approach. What the genetic research clarifies is that the variation in response between individuals is not random or unexplained. It's mechanistically grounded in well-characterized differences in synthesis and transport efficiency that simply vary across the population.